It's a pleasure and an honor to introduce our keynote speaker tonight, actually, Bryan Cullen. And I have to admit, he has a really voluminous CV, so unfortunately, it was too heavy for me to carry it, so I'm talking from memory here. When I started out in the HIV field, Bryan was already, he was already around for a while, and he's really one of the pioneers of HIV molecular biology. In fact, at that time, he was already one of the most cited authors, at least in the HIV field, according to Science, I believe, in 1993. So he made that list, and probably other times. And later, when he broadened his scope to mRNAs and things like that, he frequently was one of the, I think, 1% top cited authors in the world, actually, in any field. So he's really a big guy, and with a long and distinguished career. And in order to to illustrate this, the longevity of his career, I asked people for slides that would show him early on in his lab, I guess. And this is what I got. So I really like the outfit, by the way, coming from Bavaria. So he started out early, and then there was somewhat of a hiatus owing to the fact that he moved to the US. And by the time he got to finish his PhD in 1984, he already had around 20 papers, apparently. And that enabled him to sort of circumvent this cumbersome step of doing a postdoc. And in fact, he went right from graduating to sort of an assistant professor position, or the equivalent, at Hoffman La Roche in New Jersey. And then he quickly published a paper about HIV transactivation in cell. And Bryan was the first author in that paper, and he was also the last author. So he was, in fact, the only author, and that was very impressive. It is very impressive, and led to an offer for a Howard Hughes investigator, a position at Duke University. And Bryan chose to accept this offer, and he has been at Duke University ever since. And in 2004, he became the founding director of the Duke University Center for Virology. He also is a James B. Duke professor, which, according to my research, Wikipedia-based, is bestowed upon a very, very select group of faculty that includes 2 Nobel laureates. Very impressive. So Bryan's work, broadly speaking, has focused on RNA-mediated regulation of everything, basically. And he has made a number of very notable discoveries, such as, for example, in the HIV field, he discovered and named the Rev-responsive element. He also demonstrated that Rev is a sequence-specific RNA export factor. Likewise, that TAT is a sequence-specific transcription factor, as well as many other things. Then he, as I said, he broadened his scope, and he moved to mRNAs, or RNAi, where he had also a very big impact, particularly in the discovery of virally encoded mRNAs, especially in herpes viruses. And he was the first to discover these mRNAs in several of these herpes viruses, including, I believe, an elephant herpes virus. So recently, he actually went back to HIV, using it as a model system to unravel the roles of a post-transcriptional post-transcriptional RNA modifications in viral replication. Now, I should mention that Bryan also has an impressive track record as a mentor. So alumni from his, or notable alumni from his lab include the guy here on the right, Mike Malim, Paul Bienasch, and also I think Joachim Hauber, who is the head of the Heinrich Pette Institute in Germany. And so he has also received numerous awards and honors, which is really not very surprising. So, for example, just to name one, he appears to have gotten something like the Oscar, perhaps. and yeah, exactly. And Mario Stevenson expects something similar, apparently. Anyway, so we are really fortunate that tonight Bryan is going to tell us about his latest insights, or his insights in general, into the regulation of HIV gene expression and replication. Over to you.
That's a very good question. So, does that work okay? Oh, great. Thanks, Heinrich. I really want to thank Heinrich Göttlinger and Claire Jolly for the honor of this opportunity, which actually does mean a great deal to me. So Heinrich told me that I should do a little bit of a quick run-through of some of my earlier career, although at the risk of being slightly redundant with what he's already said. So quickly, I grew up in the UK. My mother was German, which is why I was wearing lederhosen in that picture, but that was taken in the UK. I did a bachelor's degree at Warwick University, and just to show what I used to look like then. This is my admissions picture from 1970. So I haven't changed an iota, right? Moving along quickly, I did a master's degree at the University of Birmingham in the UK, then moved to the US, where I got a job as a technician at Hoffmann-La Roche. And I'm not really going to talk about this period except to say that it was extremely a period of considerable growth. I worked in 3 different laboratories as a technician, and I learned a vast number of techniques, which stood me in extremely good stead when I did my PhD. And one of the odd things about my career is that I actually did my PhD at Rutgers while fully employed at Hoffmann-La Roche. So I was driving back and forth all day, every day to take classes, then come back and do research at Roche. And that was, in fact, due to 2 individuals. Most importantly, my graduate student mentor at that time was Grace Jew, who was actually a postdoc, had been a postdoc in Anne Skalka's laboratory. Anne Skalka was at Roche at that time, although at that time, Grace Jew and I were not in her department. Later on, she actually took over the department, and so I ended up being in Anne Skalka's department, and Grace Jew and I did a number of collaborative papers with her. So I just want to point out this particular paper as the first one I think that's notable that I was involved with. It was an article in Nature that showed that transcription from retroviral 5' LTRs inhibits transcription from 3' LTRs in cis. So the RNA polymerase II runs over the 3' LTR and occludes it to a degree. And we call this transcriptional interference, and that term really caught on and was extensively followed up in the literature by people like Nick Proudfoot and others. And so this phrase, transcriptional interference, coined in this manuscript, has actually been cited in over 6,000 papers. This was kind of the thing that started me moving in the direction of being noticed. And the part that made even a bigger impact was when I came back, and as as Heinrich said, when I finished my PhD, because I'd never actually left Roche, they simply promoted me from technician to lab head, so cutting out all this annoying postdoc stuff that a lot of people have to deal with. And at that point, I was in Ann Skalka's department as an independent researcher. And one of the things that I was allowed to do was to work on HIV-1. And the first paper that I had, because I just founded my own lab, and I actually had nobody in my lab except me. So that's why I ended up publishing this cell paper as a solo authorship. So the context of this paper, which it says occurs via a bimodal mechanism, was that it had been argued by a group at Harvard that the TAT protein was entirely a translational activator of HIV-1 gene expression. And so what we showed in this paper was that there actually was a dramatic increase in the amount of RNA expression from HIV. Now, it turned out, and this was subsequently confirmed by people like Mike Matthews and George Pavlakis, that the increase in the amount of RNA is in fact not quite as high as the increase in the amount of translation. So you could argue that there was some kind of a secondary phenotype. The important point was it looked like transcription. And at this point, Yogi Halber, that was mentioned by Heinrich, joined my laboratory as my first postdoc. So I was very fortunate to have him come to my group at what was then Roche. And this is Yogi many years later. I couldn't, you don't have digital photos from those days, so it's hard to dig them up. So he was a great guy to have joined my lab, and we actually showed in this paper that TAT was a transcriptional activator of the HIV-1 LTR. What we missed was that it actually acted at the level of elongation, as was subsequently shown by Mattia Pieterlin. And so this was actually interesting. I went to a Keystone conference and I presented this work, and Mattia, who I knew somewhat at the time, who's become a good friend, was in the audience, and he said, well, we did the same experiment using transcriptional run-on, and we got exactly the opposite result. We don't see any difference in the amount of transcription from the HIV LTR in the presence and absence of TAT. And I said, well, here's the data. We did it. There it is. And so we talked about what the differences were, and it turned out that the probe that we used for transcriptional run-on was quite a long way from the LTR, whereas the probe that Mattia had used for transcription and run-on was right at the beginning of the provirus. And so he very nicely followed that up with the Nature paper that then showed that what was actually going on was that TAT was activating transcription elongation, which downstream looks like transcription initiation. And so the data are correct, but the conclusion was perhaps not totally right. Nevertheless, undiscouraged. So the two papers, basically, the Nature paper and the Cell paper particularly, but also the PNAS paper, brought me to the attention of Duke University, who recruited me there as an HHMI assistant investigator in 1987. And what happened then was that I was extremely fortunate to get a really outstanding scientist to join my laboratory. And I think the hardest things when you're starting out as an assistant professor is to get people, especially postdocs, to come to your lab. Because there are lots of other labs with better known scientists that have seem much more attractive. So, you'll have to ask Mike why he decided to come to my lab, but it was a hell of a good thing for me. So the big thing that we did was this paper, which I think, it showed that HIV-1 is a nuclear mRNA export factor, which was the very first to be identified anywhere, and also defined and named the Rev response element, not Rev responsive element, please. Rev response element, and this was arguably the most important paper I ever published, although it only ranks as number 3 on my greatest hits list. But I talked to Mike about that, and he feels this is also extremely unjust, and that I think it's because it's an older paper, and that newer papers tend to get cited more because there are more journals to cite them, basically. But that was the first step in what was an extraordinarily successful collaboration between Mike and I. And I only had 2 postdocs in tech in the lab, and I didn't have any teaching responsibilities. I was a Howard Hughes investigator, so I didn't have to write any grants. And so I was full-time in the lab, and so it was really not so much me running a lab as the 4 of us trying to work together to do the best we could. So the next 4.5 years when Mike Malin was in my lab was really an extraordinarily productive period. This was an important paper that where we initially identified a transdominant repressive Rev function. We gave it the name M10, which became quite a well-known mutant. And what we then subsequently did was we demonstrated that there was a motif towards the carboxy terminus of Rev, which we termed the activation domain or leucine motif. And Mike then went on to show that that was a nuclear export signal. And then Matai and others eventually showed that it was the binding site for the cellular RNA export or nuclear export factor, CREM1. So this was a very important paper in moving the field along, but also was very interesting at the time as it was a very strong inhibitor of HIV-1. And it was actually shown by a group from Sandoz that if you put this into lymphocytes and put it into people, that it would protect them to a significant degree. And then we also published a whole bunch of other papers in Cell and what have you. And Mike had a hell of a good run in my lab. He was in my lab for 4.5 years. He got 22 papers, including 3 first-author Cell and 2 first-author Nature papers. So it wasn't a surprise that he never actually had to apply for a job. People were just calling him every day, "Would you come and work for us, please?" Yeah. So eventually he took a Hughes position at Penn, where he did extremely well. I didn't even mention that we actually had several envelope papers at the time too, which I don't think are cited that much, but one of them was the first to show that the V3 loop was the dominant determinant of tropism for the envelope. This is before CXCR4 and CCR5 were even discovered. And it wasn't just Rev that Mike and I were into. This is a paper that I think was extremely important, perhaps even more important than we realized at the time, because it was the first paper to show that HIV-1, unlike MLV and ALV, has the ability to infect non-dividing cells, including primary macrophages. And this, at the time, really overturned what was a dogma in the field of retrovirology. And we had a hell of a time getting this sucker published, because the reviewers kept asking for more and more and more data, and we kept generating more and more data, and they still wouldn't believe us because they fundamentally did not believe that retrovirus could infect non-dividing cells. But it stood the test of time, no question. So one of the biggest mistakes I ever made was not patenting HIV-1 as a vector at this point in time, because I never thought that anybody would use a pathogenic virus like HIV-1 as a vector, and that turned out to be a stupid error on my part. So Mike left, but other people came to my lab that turned out to be also extremely talented, and 2 that I particularly want to highlight are on this manuscript here. So this manuscript was part of the work that Paul Bianash did when he was in my laboratory from 97 to 2000. And what it shows, well, what had been shown by Kathy Jones's laboratory is that the cofactor for HIV-1 is cyclin T1, also called positive transcription elongation factor B, and that TAT directly interacts with that. And we had shown, me and Paul, had shown that the reason that TAT doesn't function in mice is that cyclin T1 in mice doesn't interact with TAT effectively. And if you change one amino acid in the mouse cyclin T1, it then becomes fully competent to support TAT function. But then the question was, does TAT do anything else? Is the only thing it does recruit cyclin T1? And what we show here is that if you recruit cyclin T1, so there's no TAR, there's no TAT, all there is an RNA element that we're recruiting cyclin T1 to in the HIV-1 LTR, it totally and completely reconstitutes TAT function. So despite any rumors that you've heard to the contrary, there is only one protein that is recruited by TAT to the HIV LTR, and that's the cyclin T1. Everything else is recruited indirectly through cyclin T1. The other person I want to particularly mention, because he's been so incredibly important in my career, is Hal Bogert. So Hal Bogert is a senior scientist in my laboratory that's been with me since 1992, through sort of thick and thin. Extremely talented scientist, has over 40 manuscripts in my laboratory, including many, many first author papers, and continues to be a person that I rely on a daily basis to help me run all the projects. And I think at this point, he knows a lot more about running my lab than I do. At this point, I actually started to move away from HIV-1. So for many of the younger people here, you may think, not think of me as a retrovirologist. And what we actually got into initially was the whole idea of RNA interference and microRNA. So this paper here in Molecular Cell was the first paper to ever express a microRNA in a mammalian cell, ever express microRNA mutants in a mammalian cell, ever generate indicator constructs for microRNAs that were active in a mammalian cell. And that turned out to be extremely important, if nothing else, because it having gotten somewhat smarter since the time that I didn't patent lentiviral vectors, I did get a series of patents which cover every single vector that expresses microRNAs or any derivative of a microRNA. So that turned out to be very useful. This one here was another one that we did at the time because we were interested, of course, in nuclear RNA export. And we showed that export in 5 mediates pre-microRNA export. And that turns out to be my most highly cited paper, which is interesting. I don't think it's by any means the best thing we ever did, but it gets cited one hell of a lot, as it says here. Finally, I've been a James B. Duke professor since 2005, and a lot of the time that I was working in that area, I was working on non-HIV-1 microRNAs. So I got into the whole idea of microRNAs thinking that viruses would use microRNAs to regulate host cell innate immune responses. It would be a nice convenient way of turning off genes that viruses don't like. You could make one microRNA, very, very small, very specific. Turns out that, in fact, they do exist in viruses, but they only exist in significant numbers in herpes viruses. And this is two very talented female postdocs from this period. Eva Gottwein, who went on to become a faculty member at Northwestern, and Jen Umbach, who went into industry after she left my lab. And this particular one describes the very first example of a viral microRNA that is simply an ortholog of a cellular microRNA that is absolutely required for transformation of cells by KSHV. And this one is the first paper identifying a whole series of herpes simplex virus microRNAs, which were unknown previously, and showing that they regulate viral RNAs in essentially antisense mechanism. And so that's the end of that. And what I'd like to turn to now is the stuff that we've been doing more recently, and hopefully you'll find that of interest. And this all deals with HIV-1 predominantly. So viral and cellular mRNAs are subject to post-transcriptional modifications. So several covalent modifications of individual nucleotides have been reported at levels over 0.01%, which is actually not very much, but it's significant. Of these, the most common is the addition of a methyl group to the N6 position of adenosine, which is called M6A. And the presence of a high level of M6A in viral mRNAs was first reported by Bob Krug for influenza virus in '76, and then subsequently for a range of viruses, including avian retroviruses, by Karen Beeman's lab in 1984. But at that point, it wasn't possible to map these, and it wasn't possible to determine what they did. Nor was it known what proteins were involved in this whole process. So work from a lot of groups, Sami Jaffrey, Chuan He, and others, subsequently revealed that the M6A modification is added by a set of proteins, metal 3, metal 14, and WTAP, that actually co-transcriptionally adds M6A to RNA in the nucleus. Those M6As can then be read by a nuclear protein, particularly YTHTC1, which could affect RNA splicing. But more importantly, they're recognized in the cytoplasm by 3 proteins called YTHDF1, 2, and 3 that can regulate RNA stability and function. And so the addition of M6A is actually separated quite dramatically. These are overexposed to make the point clearly. The 3 proteins that add M6A are in the nucleus. The 3 proteins that recognize M6A and execute its function are cytoplasmic. So one of the things we wanted to get into initially was the mapping of M6A sites. And we had previously done a lot of this kind of mapping data for microRNAs. And what we use is a technology developed by Tom Tuschl's lab, where you pulse cells with 4-thiouridine, you then UV cross-link the cells, which specifically cross-links proteins to RNAs. Doesn't cause any other cross-links. You can then isolate in this case it's MRISC, but it could be any protein bound to an RNA by immunoprecipitation. And then you can protease away the RNA that's not protected by the protein, clone the small pieces that are protected, which are about 20 nucleotides in size, do deep sequencing to identify locations where the M6A is on the RNA. And to do this, of course, for M6A, you would use one of the YTHDF proteins. There's a second protocol, which is also very useful, where you also pulse cells with 4-thiouridine, but now you isolate the RNA from the cells, and you take the purified RNA and add an antibody, which is specific for the modification. You cross-link the antibody, then again you hydrolyze away the non-bound RNA and clone and sequence the binding sites. And so if you do this with HIV-1 for M6A, and so this is using the antibody procedure in the top lane. The second lane is a negative control. And then the other three are three different cell lines, each of which expresses either YTHDF1, 2, or 3. And what you can see with this, first of all, is that almost all of the binding is in the 3' end of the virus. And then again, if you look down here, you can see that there's very, very good reproducibility, especially between the YTHDF proteins. What you end up with is an MVREV overlap peak, a NEF peak, a peak in the U3 region, and one in TAR. And all of these peaks are also seen if you look with antibodies, although the relative intensity is a little different. So one of the things we were interested in is whether these sites are conserved. And so we looked in BAL, and what you can see is that if you look in BAL, the MVREF site is conserved, the TAR site is conserved, the EU3 NF-kappa B site is conserved, the site in NEF is conserved, but now you actually have an additional site in BAL that is not found in NL43. And similarly, if you look in GRCSF, this peak now becomes broader. These two peaks are still here. This peak in BAL is not observed, and this peak is retained. And what it actually turns out is that, well, we know that the M6A consensus site is DRACH, where R is purine, but the R is almost invariably G, rarely A, and the D is basically anything but U, H is anything but G. Not much of a consensus, but it's what we have. But what's interesting is that this peak here, the BAL peak, this G in NL43 becomes an A in the consensus, and that generates the new peak. And then if you look at GRCSF, what we actually have is two new peaks, a new peak here, which is generated because you have an A in the consensus, which is quite different from what you see in NL43. And then here you have a G in front of the A instead of an A in front of the A. And I told you that G is preferred. So for this peak, it actually goes from detectable to strong. But bottom line is that sites of M6A addition are conserved across HIV isolates and appear to be present on all viral transcripts because they're in the U3 region, basically. And the question is, does this mean anything? So one of the initial experiments we did was to simply take, it's actually very hard to make mutants in HIV-1 because there's so many layered levels of information that you don't know that you're going to not mutate more than one thing at a time, even though you're trying to put silent mutations in. And so we thought, let's just cut to the chase, and we'll just take the back end of the HIV-1 provirus and make it the 3' UTR of an indicator gene. And so we either took the whole thing with all of these sites, or this region with just the U3 and TAR sites. And then we mutated the various A's that are indicated here. And then in the indicators, we asked whether the mutants expressed as much as the wild type. And you can see that for all of the indicator constructs, and whether we're in CMSS T cells or 293T, if we took the M6As out of the U3 region, we now lost expression. So a different way to look at this would be to ask whether the M6A machinery promotes HIV-1 replication. If yes, then overexpression of YTHDF reader proteins, the ones that actually detect M6A, and remember there are lots of M6A sites on cellular RNAs, so there's presumably some competition there, should enhance HIV replication, while knockout of YTHDF genes using CRISPR-Cas should repress HIV-1 replication, because M6A is no longer being read by anything, so how can it exert a phenotype? So if you actually look in T cells, YTHDF2 is the only protein expressed at significant levels. In our hands, YTHDF3 is undetectable, and YTHDF1 is very low, so we focused on YTHDF2, and we made a knockout using CRISPR-Cas, so we overexpressed it using a lentiviral vector. It's actually hard to overexpress. The cells don't really like having overexpressed YTHDF2. But the bottom line, maybe just look at this one panel down on the right. If we overexpress YTHDF2, this is the amount of gene expression that we get from the provirus in terms of NEF and P24. If we knock it out, this is the level of expression we get, and this is the starting point. So yes, indeed, if you overexpress YTHDF2, you get more virus replication, and you also get more cell killing when compared to the wild type, and even more so the knockdown. And So the problem with all this kind of stuff is, you're mapping these sites onto the viral provirus, but you don't know what it means. So you have a peak. Is that 10% occupancy by M6A? Is that 50% occupancy? There's no way to tell. This is just concluding what I just said. So the hypothesis then is, so our mapping sites identify sites of M6A addition, but they don't provide any information about the level of methylation. However, if we believe that M6A residues in cis indeed promote HIV-1 gene expression and replication, then M6A residues should be overrepresented on HIV-1 transcripts when compared to the average cellular mRNA. If they help the virus is going to accumulate additional M6As when compared to cellular RNAs that are not under the same selective pressure. So to test this hypothesis, we therefore wish to quantify the number of M6A residues per HIV-1 genome. Moreover, other epitranscriptomic modifications might also affect HIV-1 RNA function, so we wish to see if other modifications are also over or underrepresented on HIV. So what we did was we took CM cells, and I mean a lot of CM cells, infected them with HIV, isolated the virus by putting it, first of all, through a sucrose cushion, then isolated it on an iodixonal gradient. We then take the purified virus. We isolate the RNA from the purified virus. We run it on a preparative TBE agarose gel and actually visualize the HIV-1 genome as a band on the gel, excise it, and then we do ultra-high performance liquid chromatography tandem MS, which allows us to quantify exactly what modifications are present. Now, we did this with two different samples. We used CM cells. And what we did was we took the sample before we analyzed it, and we performed RNA-seq using a couple of different polymerases, both regular MLV reverse transcriptase as well as a reverse transcriptase, which has been proposed to detect any modifications that might be present on RNAs and reverse transcribe them appropriately. And from the CM, we have 96% HIV-1 RNA. We have about 4% cellular mRNAs, and we have less than 0.1% non-coding RNA and less than 0.001% tRNA, which is the most highly modified cellular RNA. 293T, we get very similar data. So then we take poly-A plus purified RNA. So we take cellular RNA, we do 2 poly-A purifications, and then we do a ribo depletion to get it as pure as we can. And then we compare the level of modifications on the cellular RNAs from CM and 293T versus from the HIV. What you will see is that, so CM is 2'-O-methyl cytosine, 2'-O-methyl guanosine, 2'-O-methyl adenosine, 5-methyl cytosine, 6-methyl adenosine, M1G and M7G, again, obvious what those are. Let's just sort of ignore these guys. But what you can see is that for every one of these modifications, you see that the viral RNAs are much more heavily modified, and this is a log scale, than the cellular RNAs. So for the 2'-O-methyl modifications, it's about 20 to 30-fold higher than a regular cellular RNA. For M5C, it's about 25-fold higher. You'll notice that the two different cellular RNAs are very similar to each other, and the two different viral RNA preps are always very similar to one another. Now, M6A, which I just showed you actually is a positive actor, is one of the least significant differences. It's only 2.5-fold more, but at least it's more. And then M7G was recently shown to enhance translation in cis when added at internal locations in RNA, and again is overrepresented. Nobody knows what M1G and these other guys do. If we actually quantify the number of modifications per RNA, I don't want to read through this entire thing, but what you can see is we get good agreement between 293T and T cells, and there are quite a lot of M6A modifications and quite a lot of M5C modifications. So 13 versus 12, 11 versus 23. So these data reveal that HIV-1 transcripts bear a far higher level of epitranscriptomic modification than does the average cellular RNA. This is from about 20-fold for 2'-O-methyl and for M5C, implying that these modifications have been selected for during HIV evolution. The virus doesn't pick up 20 times more than the cell by accident. So then the question is, what do they do? So we decided to focus on M5C next because it was so heavily overrepresented. And so for this purpose, we modified our antibody detection procedure by substituting a commercially available M5C antibody to to detect these modifications. So this has never been done. So we first wanted to check whether it actually worked. And so an M5C has been mapped on tRNA serine, and we map it to exactly where it's supposed to be. An M5C has been mapped on the 5S ribosomal RNA. We map it to exactly where it's supposed to be. It's previously been published that M5Cs are focused around the translational initiation codon. So this is bulk cellular RNA all analyzed together. And so you can see there is quite a lot of M5C right around the AUG. And here it's sort of fine mapping, and you can see again there's a lot right around the AUG and a little bit around the termination codon. We don't come up with a good consensus sequence for addition of M5C to RNA. About 20% of sites have this consensus, which is extremely significant. But that only applies to 20% of the sites. Now, there are actually 8 enzymes in human cells that add M5C to RNA. There are 7 enzymes that belong to what's called the ensun family, and 1 enzyme which is actually a repurposed DNA cytidine methyltransferase called DNMT2. Just to give you a sense of where we are with HIV-1, so the total number of M5C sites on the entire T-cell genome transcriptome is 1,641. The total of M5C plus RNAs is 1,489. So that means that for the cellular RNAs that have M5C is an average of 1.1 per RNA. But something like 70% of all of the RNA of the cells don't have any M5Cs at all. If you actually look at what doesn't have M5Cs, very, very few housekeeping genes have M5C. So here GAPDH, HPRT, beta-actin have no M5Cs at all. These are the 5 RNAs in the T cells with the most M5Cs. This one here, FAM230A, is actually a long non-coding RNA. And then these are, will come up again later, but these are just have multiple M5Cs. But even the most highly modified mRNA has a lot fewer M5Cs than the HIV-1 genome does. So we did the mapping. And I'm actually going to show you a result here which I don't know how to interpret, but you know, it's interesting. So in the previous piece about M6A, I talked about M6A being down at the 3' UTR. And all of the data we generated originally on M6A was looking at intracellular viral RNAs. It turns out when we look at viral RNAs, these same peaks, you can see they line up beautifully. All of these peaks in NAF and the MVREV overlap region, et cetera. But we have substantially more M6A on the genomic RNA than we had on intracellular RNAs, more than can be accounted for by the fact that the intracellular RNAs are spliced and the viral RNA is not spliced. And then if we look at M5C virion RNAs, they tend to be scattered around. Some of them are near ATG, some of them are not. Again, we see more towards the 3' UTR. We don't see this effect of having more modifications in the genomic RNA than the intracellular with M5C. No idea what that means. So these data indicate that HIV-1 transcripts bear multiple M5C residues. We've mapped these residues, and our next goal was then to define the functional significance of the residues. Again, we have the problem of mutagenesis is problematic in HIV-1. So while mammalian cells express 7 cytidine methyltransferases belonging to the ensun protein family, ensun2 has been reported to be the major M5C writer on mRNA, so there are 2 papers saying that. And what's really cool about the ensun methyltransferases is they have this odd property in that they form a transient covalent bond with the cytidine that they're modifying. So they bind to the cytidine, they covalently link themselves to the cytidine, and then that linkage is hydrolyzed, and during the hydrolysis step, the methyl group is added. And so you can actually mutate a particular, I think it might be in the next figure, you can mutate a particular cysteine residue in all of the ensun proteins, and what happens is that the ensun protein got stuck to the cytidine that it's supposed to be modifying. And so we reasoned if that was true, then thinking that it was ensun2, if we made this mutant, then the ensun2 protein should try to modify the genomic RNA of HIV-1 and then would not be able to release. It would be permanently stuck. And that should mean it would be packaged into the viral particle, whereas the wild-type ensun protein would add the M5C and then release. And so we expressed a number of these different ensun proteins in cells, and then looked in virion particles. These are all the mutants. And as you can see, ensign-2 is indeed extremely efficiently packaged into viral particles when it's this point mutant that causes crosslinks, but none of the other ones are detected. We used APOBEC 3G as our positive control, and you can see the level of packaging is comparable. So this opens up the possibility then that we can actually use the ensign-2 with the point mutation to map where the methyls are added to the genome of the virus, because they're going to cross-link without any UV light or anything like that. And it turns out that that's exactly the case. So if you look at ensign-2 wild type, you basically see very little binding to the HIV-1 RNA intracellularly. If we look at this point mutant, we get a lot of binding, and the binding sites line up very nicely with the binding sites measured by PAM5C-seq using the antibody. So this is another line of evidence that there are a lot of M5Cs on the HIV-1 genome. The other thing, of course, you can do is you can knock out ensign-2 and then ask, do the M5Cs go away? And it turns out, if you compare this lane to this lane, that almost all of the sites do go away, except for 3. 1, 2, 3 sites are completely unchanged when you knock out ensign-2. So what that tells us is that the vast majority of M5Cs on the HIV-1 genome are added by ensign-2, and that 3 sites are added by somebody else, and we don't know who that is. So what happens when we knock out ensign-2? Well, what happens is that the virus grows less well. If we put the ensign-2 back, we recover. This is looking at NLUC in place of NEF. This is looking at GAG expression. So in the absence of ensign-2 and in the presence of ensign-2, you see this significantly higher level of production. So following this a little bit further, we can take 293 T cells and we can transfect them with a proviral indicator plasmid that produces infectious HIV-1, and we can co-transfect wild-type ensign-2 or the dominant negative mutant or the mutant that cross-links or another mutant which is unable to interact with RNA. And you can see that when we put the wild-type back, we get significantly more GAG production. If we now look at the level of NLUC production from this vector, it closely mirrors the amount of GAG production from this vector. And if we look at the amount of mRNA produced in these cells, it's completely unaffected by the presence or absence of ensign-2. So ensign-2 is increasing viral protein production without increasing viral RNA production. If we look at the virus supernatant from these cells, we see that there's actually significantly more infectious virus produced in the presence of ensign-2 than in the absence of ensign-2. Again, we see that 271A, the mutant that cross-links, is in the viral particles, but the others don't get in. And I think I'll just miss out that last part. Now, we did a lot of work on Rev, and Rev was first proposed to be a translation factor. And the reason is that if you look in cells that do or don't have Rev, the amount of HIV-1 RNA tends to be pretty similar. And so we were very aware of the fact that a translational phenotype could actually be a nuclear cytoplasmic distribution phenotype. And so we wanted to know if maybe ensign-2 is increasing the nuclear export of HIV-1 RNA. Very unlikely, because we know that Rev does that, and it doesn't presumably need any help. So these are just all kinds of controls for fractionation. Ensign-2 is going to be nuclear, LMN is also nuclear, laminin, and GAPDH is cytoplasmic. And if you look at the RNA localization in the absence or presence of ensign-2, there is no difference whatsoever. Now, just to remind you of this slide, so here's HIV-1 with 11 M5Cs, and here's MRAS with 5 M5Cs, and here are 3 housekeeping genes with no M5Cs. When we knock out ensign-2, if we believe that translation is being affected, then we would predict that the RNAs that have lots of M5Cs should have a lower association with ribosomes, whereas the ones that have no M5Cs should be unaffected. So that's our control for that it's not some kind of global phenotype. And that's exactly what we see. So you can see we look at ribosome loading. In the absence of ensign-2, the GAG RNA is substantially decreased. The cellular MRAS RNA, which has 5 M5Cs, is substantially decreased. These are all set to 1. Doesn't mean that this is more efficient or less efficient translation. But the 3 housekeeping genes, GAPDH, actin, and HPRT, are unaffected. And so the presence of M5Cs in cis increases the ability of mRNAs to be recruited to ribosomes and to be translated. One of the things we did in collaboration with Ron Swanstrom and Anne Emery is to ask whether there was any effect on splicing, and we're all colleagues in the cRNA operation that Alice Telesnitsky runs, and so it's nice to have the opportunity to collaborate with people who are in that same group. And so we did all the relative transfections, and then we sent all the RNA over to Anne, and she did her analysis, which is an extensive deep sequencing analysis to look for different splicing events. And what was interesting is if you knocked out NSON2 in blue, the splicing event from the major donor D1 at the beginning of GAG to A2, which is the middle of VIF, was substantially inhibited, whereas all of the other splicing events were largely unaffected, and that was independent of whether you measured the 1.8 kb class of RNAs or the 4 kb class of viral RNAs. So what was interesting then is that there was an M5C site just upstream of the A2 splice acceptor detected both by N2C271A clip, so the NSON protein that cross-links, and by antibody recognition. And we can knock this one out, but not in a replication-competent clone because we can't make silent mutations and get rid of all the Cs. But in the context of 293Ts that are transfected, it doesn't matter. So we got rid of the Cs, as shown here. There are actually 6 of them in this peak. And what happens is that the mutant then largely recapitulates, as you can see here, the NSON2 knockout phenotype. So the NSON2 knockout phenotype is going from this black bar to this black bar. If you make the mutant, it goes from this black bar down to here, and you have no significant additional inhibition when you knock out NSON2. So we can get the same phenotype in cis and in trans. So HIV-1 RNAs are heavily M5C modified. NSON2 is the major M5C writer for HIV-1 mRNAs and the 271A NSON2 mutant spontaneously cross-links to HIV-1 transcripts. This can be used to map NSON2 specific M5C modifications and results in NSON2 incorporation into HIV-1 virions. When we knock out NSON2 by gene editing, we get lower HIV-1 gene expression and virion production. And this inhibition is further exacerbated. I didn't show you this, but if we take the NSON2 271A mutant, which cross-links to the HIV-1 RNA, of course it screws up reverse transcription, nothing too surprising there. So M5C's residues promote HIV-1 gene expression, at least in part by enhancing viral mRNA translation. And just as an aside, this is a paper that came out very recently from the Benasser lab, and she showed that the addition of 2'-O-methyl groups to HIV-1 allows HIV-1 to avoid innate immune sensing. So I told you that 2'-O-methyl modifications are 20 to 30-fold higher on HIV-1 than they are on cellular mRNAs. And so we have another example of a modification that appears to be promoting viral gene expression. So basically, every modification that we're able to assay except inosine is up on viral RNAs. Do I have like 5 more minutes? Yeah. So this is a retrovirus meeting, so I thought I'd throw in some MLV data. And basically, it's more or less the same data, but done with MLV, except with MLV, you can get really, really pure RNA. So we have 3 separate genomic RNA samples from MLV virions, all of which are 99.5% or more purified. And when we look at the various modifications and compare the level of modifications from MLV genomic RNA to 3T3 poly A plus RNA, what we see is, again, this huge excess of modifications in the MLV RNA relative to the genomic RNA of MLV relative to the cell. And in this particular virus, actually we see a big increase in M6A, almost a 10-fold increase, which we didn't really see with HIV. But again, with 2'-O-methyl C, G, and A, there is a massive excess. And M5C is about a 30-fold excess. And again, we can map the M6A and M5C peaks, and we can actually, the nice thing about Maloney virus is a lot simpler. So you can mutate things with at least a little bit of confidence that you're not hitting something you didn't know was there, because there's only one splice donor and one splice acceptor, and not much else. And so we can actually then put specific point mutations into peaks. And just as an aside, this is the absolute number of these residues. So if you look at M6A, it actually has 51 M6A residues per MLV genome, which is a lot of A residues for a viral genome. And some of the other ones are there at very high levels as well. And they're very comparable in many ways to what you see with HIV. I mean, 27 versus 30 3, 3, 4, 4, 25, 21, etc. But anyway, looking at these mutants, this one which has some M6A sites mutated and this one which has some M5C sites mutated, the bottom line is that if you look at their ability to replicate, it's reduced by 2 to 3-fold. And if you take the supernatant from these cells, you can see substantially lower levels of viruses being produced. So this is now a cis phenotype as opposed to a trans phenotype, but we can get the trans phenotype too. So if we overexpress mouse YTHDF2 in 3T3 cells, we get better production of P65 GAG compared to the wild type cells. So we're not comparing overexpression and knockout, we're just comparing overexpression to wild type. Again, we can get NSUD2 is packaged into MLV virions, arguing that it's going to be the protein that adds M5C to MLV genomic RNAs. And if we knock out NSUD2 in 3T3 cells, we see a substantial drop in the level of MLV gene expression as well. So that's it, and I don't have pictures of the people that are in my lab now. David Courtney just left my lab. Very, very talented postdoc who did all of the work on M5C and HIV1 and MLV. Matt Kennedy was a postdoc in my lab that did the M6A work with HIV1 that left to go to Oncorus. Hal Bogart helped out with everything, and Kevin Tsai helped out with some of the work with the M5C work and M6A work. And Anand and Ishak have been in the lab just for a short time, and as has Heather. Well, this is work on influenza virus that I didn't talk about. But Chris Holley is the person at Duke who does the UPLC tandem MS work for us and his student Brittany Law. And the splicing work with M5C was done by Ron Swanstrom, or specifically by Anne Emery in Ron Swanstrom's lab. And that's all I got.
Well, that was fascinating. Questions for Bryan?
That's beautiful. Any idea about what the readers are for M5C that improves translation elongation?
Yeah, we thought we had it. We thought we had it. We did a lot of work on that, and we ended up identifying a protein called MBD2, which is actually a protein that recognizes 5-methyl-deoxycytidine. And when we did IF, it was partly in the cytoplasm and partly in the nucleus, and then we did a lot more work, and then it all sort of fell apart. It was basically we were looking at a protein that binds deoxy M5C, and it could bind ribo M5C at a lower affinity, but still with some specificity. So, bottom line is nobody knows what the reader is for any modification except M6A. That's the only one it's known for.
Hi, here.
Yeah.
Nice talk. 2 quick questions. First is about the M5C. You mentioned there are 8 enzymes as a writer. I wonder if you compared the expression profile in CD4 T cell, like NSUN2 you mentioned as a major focus.
Yeah, we didn't do some of the NSUNs. So some of the enzymes are cytoplasmic, some of them are nuclear, a couple of them are mitochondrial, and therefore presumably are not of interest. So the other one that's been proposed to modify mRNAs is DNMT2. We knocked out DNMT2, and it had no effect whatsoever on the M5C addition profile of viral RNA. So that's not the one that does the extra 3. Because we know that NSUN2 does all of them except those 3. And that's the only one that's really been shown by other groups. Nobody's looked at viruses. There's no papers on M5C and viruses have been published. But a couple of people have argued that M5C is added to mRNA by NSUN2, and our data would certainly agree with that work. Yeah, obviously another one of the NSUNs, or maybe even more than one of the NSUNs, is adding M5Cs a little bit, but none of them at the levels of expression that we have packaged into viral particles. But if there are only 3 peaks, that would be a very small number of molecules, even if it was 100% efficient, right? So we probably wouldn't see it.
Yeah, thanks. The second quick question is about the FTSG, the 2 methylation you mentioned. Would you think other modifications, like what you identified, M6A, M5C, it could be immune invasion strategy of HIV RNA?
Yes.
Thank you.
Right in front. This is gorgeous. Is there any evidence, experimental or two-carbon induced, or anything that Rev has to negotiate with these modifications? Or
Well, we know that M5C and M6A are nuclear. So the M6A, it's been shown to be co-transcriptional. M5C, it's been shown to be nuclear, but it's probably co-transcriptional, but it's not been demonstrated. For the other ones, it's not known. We have not, I mean, neither knocking out M6A or knocking out M5C has any significant effect on the nuclear cytoplasmic distribution of RNA. We haven't knocked out, I showed that FTSJ whatever paper just because it was an example of another modification that's highly overrepresented on viral RNA that's been argued to be significant. I'm not saying that they're right about what even in terms of identifying the modifying enzyme, but I think it's certainly suggestive that going forward, we will find that multiple different modifications are advantageous to the virus, and probably in different ways. Some of them might be translational, like M5C. Some of them might be more of an effect on RNA half-life, like M6A. Some of them might be avoidance of innate immune responses, like 2'-O-methyl, and it's also been shown that pseudouridine can help to avoid detection of RNA. Unfortunately, our MS system doesn't recognize U's, so all the U-modified residues just fall out of the system. We've actually recently extended this work to show, we now know that there are a lot of N-acetylcytidines on the HIV-1 genome, and a paper was recently published showing that N-acetylcytidine facilitates translation of cellular RNAs. So that's another one that's, could well be on the list. In order to detect it in the MS system, you have to have a molecular standard, so you know exactly where it runs. And we only just bought that, so we'll find that out soon. But I think this is a field that could be exploding, actually. And I hope other people will be inspired to take a look.
Brian, over here. So, I mean, you sort of alluded to it because you went into Maloney, and you mentioned flu on the last slide. Do you have a sense of how global this is across different types of RNA viruses?
Yeah, I had a couple of extra slides. One of the slides that I think I would have liked to have shown, a lot of the modification events are methylations. So M6A, M5C, 2'-O-methyl. All of those methylation events depend on S-adenosylmethionine. If you inhibit S-adenosylmethionine production using drugs that block S-adenosyl homocysteine hydrolase, and there are several drugs available that will do that, you can strongly inhibit the replication of almost every virus known to man, certainly every virus that's ever been looked at doses that are non-toxic. And so it's been shown, for example, that if you inhibit S-adenosine homocysteine hydrolase in animals at doses that don't show any toxicity in animals, you can rescue 100% of mice infected with Ebola virus and 100% of cotton rats infected with respiratory syncytial virus. To cytoplasmic RNA viruses, we've shown in a paper that we published in Cell Host and Microbe that if you remove M6A residues in cis in influenza virus, you can substantially reduce the in vivo pathogenicity of influenza virus in mice. So I think it's going to be very important for a lot of viruses, and it's possibly a real Achilles heel, especially for viruses that have short lifespans, where you could inhibit a cellular enzyme for a reasonable amount of time without causing substantial toxicity, and yet have a dramatic effect on a virus that's trying to produce tens of thousands of copies of RNA in a cell, all in one fell swoop. So there's no way I can see for the virus to get around this. I mean, it's a cellular target right now.
And maybe even the messages of DNA viruses?
We did it with SV40, and it's also inhibited. It has M6A. Actually, interestingly, not on the early transcripts, but extensively on the late transcripts, which are expressed at much higher levels than the early transcripts. And again, if we inhibit S-adenosine homocysteine hydrolase, SAH hydrolase, we substantially inhibit SV40's ability the plaque size goes down dramatically. And the cells are happy enough. We don't see any difference in cell growth or metabolism.
So, Brian, question was going to be, so with a retrovirus, you said that these marks are being put on co-transcriptionally, but it's basically a host gene. So what is specifying that this is a viral transcript during transcription? I mean, and so the question might be, do you...
Well, I don't think that...
Will a retroviral vector have the equal level of methylation, for example? I mean, is it sequence? Are you implying that...
Yeah, so depending on the modification, there's a different answer to that. So for M5C, some of the sites have that one consensus, which I assume is one of the 7 enzymes, but not the other 6, perhaps. Why some of the cysteines are picked out and not others is completely unclear. I mean, it could be structure, it could be who knows what. For M6A, it's much more of a consensus. It's always this purine, basically GAC, anything but purine, and anything but G. Obviously, that's not much of a consensus. Like there are lots and lots of sites like that, and many of them are never used, and they're not used in cellular RNAs, and they're not used on viral RNAs. And why the consensus? I mean, the consensus is necessary but not sufficient. Why that is? I don't think that the cell has any distinguishes between the virus and cellular transcripts in any meaningful way. I think that the virus has just evolved to have acquired the number of M6As that maximize the benefit to be gained from having M6As, essentially.
So very nice talk. It's known that cytosine methylation in DNA induces mutation, spontaneous deamination. Do any of these modifications in RNA induce such mutation? And if yes, could this help the virus
inosine
evolve?
does. I mean, I think that's why there's no inosine in the retroviruses, because if there was an A residue that the cell wanted to change into an inosine, it would then be reverse transcribed as a G, and it would soon become fixed as a G in the genome, right? And so I assume that's why we don't see inosine. As to whether any of the others have an effect, M6A and M5C has been known to be reverse transcribed perfectly well, as has, I mean, it's certainly known that pseudouridine is well tolerated. There's been some literature saying that M1A, and we see a couple of M1A residues, is not well tolerated by reverse transcriptase. We haven't looked at that. I mean, presumably the virus must be able to get around it somehow, otherwise it would be inhibitory, right? I mean, I think, you know, basically to quote John over here, if the virus avoids all possible things, you know, even at the single nucleotide level, you can't assume that it's not subject to selective pressure or some. If I'm misquoting you, I apologize.
I mean, if you look at the map of diversity, do you see these positions to be mutated more? I see more isolate with this.
We haven't looked at that. We do see that they're conserved, as I showed for M6A, across different isolates, right?
Brian, if I got it right, your comparison of the cellular RNA modification and viral genomic RNA modification, the source of the viral genomic RNA was from viral particles and the cellular RNA from cells. So it is known that retrovirus package cellular RNAs. How is the modification rate if you look at the cellular RNAs packaged into retroviral particles?
So, okay, we can't quantify the modification level on the cellular RNAs and the viral particle because we have to purify them to homogeneity. But what we have done is mapped the modifications on the cellular RNAs and the viral particles, and they're the same as the cellular RNAs that are in the cell. But I don't know that means that it's exactly the same level at the same location. I just know it's at the same location.
Over here. In Sammy Jaffrey's lab, he showed that, well, so the work showed that a single M6A in the 5' UTR could promote cap-independent translation. I might have this wrong, but through recruitment of EIF3. That seems like something to be really nice for a virus to do. Have you looked at cap-independent translation?
Yeah, that was actually Kate Mayer, who's a colleague of mine at Duke now. Yeah, you know, I'd rather not talk about that, frankly.
Fair enough.
But I will make the point that isn't it striking that if M6A is sufficient to initiate translation, that viruses have evolved irises of hundreds and hundreds of nucleotides to do the same thing when all they need is a single M6A?
Yeah.
It's like, and all of us here, all the virologists in the world just haven't noticed this? I mean...
That's why I mentioned it. It's pretty spectacular.
Yeah, I mean... You know, we haven't addressed it directly. I get into enough trouble as it is without, you know, going around disproving my colleagues' work. Or
Okay, any other questions? All right then, thanks Bryan for a good start.